I'm not going to say now more on this later because these are really experiments that are ongoing in the PhD thesis of Dario Sant'Antonio, who is also in the audience. But I think the three talks so far have laid the grounds of the therapy. Now I know that you want to see results. Does the therapy work in actual models of cystic fibrosis or lung infections? That's the first question, and this will be answered by Dr. Murielle Baltazar. The second question that you may have, if not, I'm going to say it to you anyway, is the therapy safe? So what happens with the healthy tissue surrounding the infection? And this will be addressed by Dr. Maddalena Mognato in the second talk. And finally, can we develop a pharmaceutically acceptable form of the therapy to deliver this therapy to the lungs? And this will be unfortunately online, but this will be given by Chen from Cardiff Scientific Line, CSL, sorry for that, Syntographics, I was going to say. And this will be the final talk this morning. So, from the ground to the applications. Murielle, please, on stage. Thank you.
Okay, thank you, Santi, for the introduction. So I'm Dr. Murielle Baltazar, and I'm postdoc at the University of Liverpool. And today I'm going to present you the bacterial models that we have developed in Liverpool to look at the efficacy of light in a more complex model. So this morning you had a very good introduction of lung infections, and you know now how they are very difficult to treat, especially in cystic fibrosis. Patients, because they produce a very thick mucus, which is concentrated and thick and very sticky. In this patient, you also have a reduced mucociliary clearance, and due to these features, patients are colonized in the respiratory tract with bacteria that can form biofilms, as you can see here. And as explained this morning, the two species commonly found in cystic fibrosis during infections are due to Staph aureus and Pseudomonas aeruginosae. So in Liverpool, we have developed a very good model to mimic the sputum in cystic fibrosis patient. So this model is an artificial sputum medium model, which uses artificial sputum medium to mimic the sputum that we can find in cystic fibrosis patient. And when this medium is inoculated with bacteria, bacteria can form very thick and sticky mucus, as we can see here, because they form microcolonies. And these biofilms are very resistant, highly resistant to antibiotics. So basically, we prepared our bacterial inoculum in ASM. Then we use this mixture to inoculate microplates. And these plates are incubated at 37 degrees for 1, 2, or 3 days, even more. And then we irradiated our biofilms with blue light. And then we disrupted the biofilms to determine the bacterial load and see if we could have a photokilling effect following light irradiation. So we perform experiments with Pseudomonas aeruginosae, and as you can see in these two graphs, we use Pseudomonas aeruginosae P01 strain or LSB65 strain, which is a cystic fibrosis isolate. And as you can see, at all time points, we had significant reductions of biofilms following the light treatment. We observed exactly the same results with biofilms produced with methicillin-resistant Staphylococcus aureus at all time point. Similar to Pseudomonas, we have a reduction of the biomass in biofilms following the light treatment. An interesting thing that I have observed when I was performing the CFU counts is that, contrary to biofilms that were not treated with light, for which I could see visible and countable colonies on plate just after an overnight growth, For biofilm treated with light, I had to wait more than 24 hours to see visible colonies, which means that following the light treatment, the bacterial growth of remaining cells in biofilms was altered. And to confirm this observation, we performed regrowth assay. So basically, we put biofilms that were not treated with light or treated with light in nutritive medium, and we looked at, over time, the bacterial growth. So for biofilms not treated with light, we saw that they were capable of growing over time, normally. But for biofilms that were treated with light, we saw that, compared to the control, we saw that they were delayed in growth over time. We calculated the area under the curve for each growth curves, and as you can see here, we observed a significant reduction of the area under the curve for biofilms that were treated with light, which means that in addition to the reduction of the bacterial load within biofilms, remaining cells are delayed in their growth, and this could potentially lead to a more successful treatment by other therapeutics such as antibiotics. And this is an experiment that we performed. So we performed a new biofilms assays, but this time, following the light treatment, either biofilms were put back at 37 degrees for 24 hours without any treatment, so they were our control, or they were treated with an antibiotics. So for biofilms grown with Pseudomonas aeruginoides, we used tobramycin, which is an antibiotic used to treat infections with Pseudomonas. And then plates were re-incubated at 37 degrees for 24 hours. And then we disrupted the biofilms to look at the bacterial density. So here are two independent experiments. So if you look at the two first bars, the blue bar and the yellow bar, that are biofilms not treated with light and biofilms treated with light, we have no differences in the biomass. Because as I told you, after the light treatment, we re-incubated biofilms for 24 hours, which means that following the light treatment, we are capable to kill bacteria, and the remaining cells are delayed in their capability to regrow, but they can regrow at some point at similar level to the one observed before the light treatment. However, if you look at biofilms not treated with light, but only with tobramycin, we could see a significant reduction, which means that the tobramycin alone was capable of killing bacteria. And this biofilm reduction was increased when biofilms were treated with light and with tobramycin. It is important to notice here that if you look at the log reduction, in the condition of light plus tobramycin, we could reach more than 2.6 log reductions, which is a huge reduction compared to biofilms that were treated with light only, for which we never obtained more than 1 log reduction. So light treatment in combination to tobramycin, in the case of Pseudomonas aeruginoides, led to a greater reduction of biofilms compared to light alone. And we also conclude that light increases the susceptibility of biofilms to antibiotics. In Liverpool, we also have the capability to work in vivo. We have different models of lungs infection in murine model. So we work with BALB/c mice, in which we can induce acute infection or chronic infections, which means that acute infection mice will be sick very quickly post-inoculation, while in chronic infections they will be chronically and stably colonized with bacteria in lungs. So for these in vivo experiments in mice, we used the Pseudomonas aeruginoides B9 strains, which is a strain which is pan drug resistant, which means that this strain is resistant to all classes of antibiotics. So mice were inoculated by, infected by intranasal dosing, and after 6 hours post-infection, mice started to develop signs of disease. Then they were culled, and we collected the lung tissues that were put in a tube containing PBS. So we did this because we really wanted to maximize the exposure of bacteria attached to tissue for the light irradiation that we perform for 50 minutes, and then we determine the bacterial load. So if you look at our first data, you see that before and after light treatment, there is no photokilling effect of the light on benign strains. If you look at the plate on the top before irradiation and the plate at the bottom after irradiation, we had similar CFU counts. So the question was, if in vivo in lungs, do bacteria become resistant to light? Why we don't see any light effect? So to respond to this question, we perform another mouse experiment where benign strain was this time pre-treated with 5-LA, no Ki, 5-LA, which is, as Sergio said, a precursor of protoporphyrin. So mice were infected, then we wait 6 hours post-infection to see the development of the lung infection. Lungs were collected, and then lungs were homogenized in PBS containing Ki alone or Ki plus 5-LA, as Ki is an enhancer of ROS and 5-LA a precursor of protoporphyrin. Then we expose tissues 50 minutes to light, and we determine the CFU count. But the first thing that we had to do was to check if Ki alone or Ki plus 5-LA had a bactericidal effect without light. So this is what we do. We look at this, the number of CFU before and after the potential incubation for 50 minutes with light, but in this case you have no light, just to see to have the control of the experiment. And we didn't observe any differences in terms of CFU counts. As you can see on the plate, we have similar bacteria before and after 50 minutes of incubation with Ki alone or Ki plus 5-LA. So we performed the same experiments, but this time our samples were exposed to light. And as you can see, with Ki alone or Ki combined with 5-LA, in both conditions, we observed a significantly great reductions of the bacterial density following the light treatment. If you look at the plate at the bottom, you can see that at the same dilution, we have a great reduction of the CFU counts, which means that a pre-treatment with Ki alone or Ki plus 5-LA can restore the susceptibility of bacteria to light. And this is a proof of concept that our protocol allowed us to kill a pan-resistant isolate. So in conclusion, so the message that you need to take with you is that the blue light induces a reduction of the bacterial density in biofilms by killing drug-resistant bacteria, by altering the bacterial growth, but also by increasing the susceptibility of bacteria to antibiotics. In addition, we showed that the light is effective to kill bacteria from lung tissues, which is very promising results for the eradication of biofilms in lungs. Thank you.